Hello, I'm Who Are We, and today we are going to dive into a paper by Lucien Tine and colleagues from 2011, which fundamentally challenges what we thought we knew about pike in Europe. For centuries, the widespread pike across the continent was considered a single species, Esox lucius, the Northern Pike. This paper, however, presents a powerful molecular and phenotypic evidence to show that there is actually two distinct species in Europe, proposing a new one, the Southern Pike, Esox Flaviae. The impetus for this research stems from conservation. Southern European pike populations have been declining, and to manage this, stocking programs often introduce pike from Northern European sources. The authors recognize that this practice, common in recreational fishing, poses a major threat to local diversity, especially if those local populations represent distinct endemic lineages. They explicitly set out to test the hypothesis that different pike phenotypes geographically isolated in Europe might actually represent two evolutionary entities. Methodology: a fusion of evidence. To address this, the team employed a rigorous multifaceted approach, combining classical taxonomy with cutting-edge molecular methods. They analyzed a large sample of pike from continental Europe, northern Italy, and central Italy. For the phenotype, they focus on two key characters: the qualitative skin color pattern, the meritistic character of number of scales in the lateral line. For the genetic signature, they used a multi-locus approach, mitochondrial DNA, using two gene regions, cytochrome oxidase subunit 1 COI and cytochrome B or CYTB. They applied the GMYC model, the generalized mixed Yule coalescent model, which is a coalescent-based approach designed to statistically detect independent evolutionary lineages, essentially providing a genetic signature of distinct species. Nuclear DNA, using AFLP, amplified fragment length polymorphism, to test overall genetic differences and the degree of historical admixture between groups across the entire genome. The discovery: two distinct lineages. The results show a clear and highly consistent separation, indicating two distinct lineages tied to geography and specific physical traits. Phenotypic separation. The distribution of phenotypes was striking. In continental Europe, the pike displayed a single dominant phenotype, the round spot RS pattern. In contrast, the Italian populations primarily showed four different patterns: stellate spot SS, diagonal bars DB, longitudinal bars LB, and vertical bars VB. The authors note that the presence of the round spot pattern in Italy is likely due to the stocking programs. The clearest diagnostic trait was the number of lateral line scales. The round spot phenotype consistently had a significantly high number of scales, generally ranging from 125 to 148. The four southern phenotypes consistently had a significantly lower number of scales, generally ranging from 101 to 115. This distinction was clear, with almost no overlap, providing a robust method for morphological discrimination. Genetic separation. Molecular data confirmed this split unequivocally. GMYC model and phylogeny for both COI and CYTB. Phylogenetic reconstruction resulted in two distinct, well-supported groups. The GMYC model provided significant statistical evidence for two independent evolving entities, essentially confirming the existence of two species. Geographic distances. Interestingly, the widespread group, the northern one, had haplotypes that were more similar to those from Canada and China than they were to the ones of the southern Italian haplotypes. This suggests that the northern pike lineage is truly a massive Paleoarctic entity. AFLP and FST. The nuclear AFLP analysis identified two distinct lineages, AFLPA and AFLPB, that were completely linked to the color patterns. The AFLPB genotype was restricted to the round spot fish, while the AFLPA genotype was exclusive to the southern barred and stellatite spotted fish. The FST value between the two groups was high enough, FST equals 0.57 for COI groups, to denote the very great differentiation between isolated populations, lending strong support for the distinct species concept. The authors also successfully identified highly informative single nucleotide polymorphisms, two AFLP fragments, band 9 and band 24, that were biunequivocally related to the southern and northern skin color patterns, offering a rapid, reliable molecular test to distinguish the two groups. Conclusions and the new species. The combined and highly concordant evidence from mtDNA, AFLP, SMPs, scale counts, and skin color patterns support a clear differentiation between widely distributed northern lineage and a geographically restricted southern lineage. The widely distributed lineage, which likely originated from continental Europe, is retained as Esox lucius sensu stricto. The southern lineage, found in Italy, is formally described as a new species, the southern pike, Esox Flaviae, NSP. The differential diagnosis for E. flavii is clear. It is distinguishable from E. lucius by its significantly lower number of scales in the lateral line, ranging from 101 to 115. Its color patterns are variable, stellate-type spot and bars, but it never displays the round spot pattern typical of E. lucius. The existence of individuals with mismatches between their mitochondrial, nuclear, and phenotypic characters in areas like Lake Maggiore and the Danube River is suggested to be evidence of hybriditism, either recent due to human stalking or historical from past connections. Practical applications for fishing. This is not just academic, it has immediate critical implications for conservation. To protect the endemic and distinct southern pike, Esox Flaviae, against genetic dilution, all programs using northern pike from other European sources for stocking in southern Europe must be immediately halted. The easy visible phenotypic markers, specifically the lower number of lateral line scales from 101 to 115, and the absence of the round spot pattern, can be utilized by local fisheries and management agencies for the rapid identification and protection of this newly recognized native species. I want to thank you all for watching. If you enjoyed this, I do have a book in the description. It goes over the systematic approach to catching northern pike. It is very scientific in this method. It combines hundreds of studies to create an amazing book. And link in the description. Thank you all for watching, and bye.